Dr. Asha Khanna. I was the HOD of this department, and the teachers over here are still very kind to me, and they call me.
विज्ञापन
आप सबको दिख रहा है slide पहला?
Yes, yes.
Okay. So now I have started a research lab of my own. I made a society for 5-6 people are with me. The name of the society is Avika Society, and the lab is Avika Biological Research Foundation over here in Nepitown, Jabalpur. Now the paper I'm presenting today, we have worked with 9 students, mostly from the Government Science College. Am I audible, please? सुनाई दे रहा सबको?
Yes, yes, yes.
Yes, ma'am. आवाज आ रही ma'am.
Okay. I worked with 9 students, and right now we have been working on the cytotoxicity of various insecticides and the ameliorative effects of plant extracts on peripheral blood lymphocytes by MTT assay and ELISA plate reader. So उसी का मैं यहाँ present करूँगी. इस बार हमने लिया है cytotoxicity of imidacloprid in vitro on lymphocytes by MTT assay and recovery by Tinosporacordifoli stem extract. Now all of you know that Tinosporacordifoli, which is known as giloe, is a very great potent promoting agent these days. तो उसको हमने ट्राई किया था कि इमिडाक्लोप्रिड से क्या हो रहा है और टिनोस्पोरा डालने से क्या हो रहा है. Okay. Now the next slide. These are the authors. Authors में I am Asha Khanna, then Manushi Mukherjee, Muskan Kachi, both from Science College, Daishankar Gautam, also from Science College, now in St. Aloysius College, Saurabh Pagare, now in Nutan College, from Science College also, and Madam Varsha Aglave. So इनके departments लिखे हुए हैं. I am a retired professor, okay, and I have directed the whole research. Now the outline is the introduction is there, in which in this introduction there's material method, then the method of preparing Tinosporas extract, then lymphocyte separation, then MTT assay itself, then trypan test of viability also, results and discussion, conclusion, acknowledgement and references.
50%
Okay.
देखो ये ले आए हैं चार्जर से वहाँ रखा है.
Now, in introduction, I can tell you that imidacloprid is an extensively used systemic insecticide. बहुत use होता है, यहाँ भी use होता है, for control of insect pests of different crops. It is preferred because it has a special affinity for insect neonicotinoid acetylcholine receptors, लेकिन vertebrate को नहीं affect करता. This is an important plus point for this. Insect के ऊपर ही affect करता. However, it tends to accumulate in the environment over long periods of time and can cause soil and water pollution. Now, because it is a foliar spray, so this study holds great importance because we have studied the toxicity of the drug in small concentrations, because spray जो कर रहा है उसके blood में तो small quantities ही जाती है ना. So whatever results we have will be helpful in formulation of safety guidelines. Okay. Then imidacloprid, this is a chemical structure of imidacloprid, and its chemical name is given. This is the chemical name. The molecular formula is C9H10, chlorine and nitrogen 5 oxygen 2, and the molecular weight is 255.6 gram per gram. Okay. Then, as its molecular weight is 255.6 grams, so the first thing we did was to prepare a 4 millimole solution. Why we have taken millimole, I'll just tell you. Was by dissolving 20.4 milligram of imidacloprid in 20 ml of distilled water. From this stock solution, we prepared 1.5 millimole, 2.5 millimole, and 3.5 millimole solutions by appropriate dilutions with distilled water. Okay. Now these concentrations, आप पूछेंगे कि इतना ही लेके क्यों चले? So before starting any research work, we have to see where to start. So we looked up and we found that Maria Elena Calderon et al. In 2011, they conducted an extensive study on different types of imidacloprid, and they found that the toxicity was ranging from 1.7 into 10 to the power minus 3 to 2.0 into 10 to the power minus 3. तो -3 हो जाता है मिलीमोल, इसलिए हमने मिलीमोल चुना. Okay, I hope I'm clear why we started with that. आप लोग को सब सुनाई दे रहा है, दिख रहा है बेटा आप सबको? हेलो?
Yes, ma'am, दिख रहा है.
Okay. I'm very elder, so I call everybody beta.
Yes, madam.
So, now Tinosporacordifoli is a woody climbing shrub native to India. आपने घरों में भी है, मेरे घर में बहुत उगा हुआ है. It has got heart-shaped leaves and it is a climber, and its stem is said to be very useful. लोग उसका पीस के काढ़ा बना के पीते हैं. But then you can even chew a raw leaf. कच्चे पत्ते को भी आप चबा सकते हैं. मैं कभी-कभी खा लेती हूँ. Okay, now this. Now Tinosporacordifoli, this is a this Giloy or Tinosporacordifoli or Guruchi, they are Himalaya capsules also. Now this is considered to be useful in various diseases: diabetes, rheumatism, anti-cholesterol, anti-arthritis, anti-hepatitis, peptic ulcer disease, and so on and so forth. बहुत सारा इनके इसके use बहुत जगह है. Okay, so that's why this was chosen and the stem was chosen. This is the stem. This is the stem. Okay. Now the aqueous extract was made. The techniques दिए हुए हैं यहाँ Sharma et al. And Sengupta et al. Now the 5 grams of the paste was taken and further ground in 20 ml of distilled water and it into a slurry, and this was kept for 4 hours in the laminar flow chamber, and after that it was filtered with Wortmann number 1 filter paper and it was used fresh. इसको फिर fresh हमने use किया था. Okay. Stem का extract. Now lymphocyte separation, there is a set procedure for this. So what is done actually is that blood from a donor is called for, a voluntary donor, and in this experiment 3 ml of the blood was taken. It was diluted in the laminar flow under sterile conditions. Then that blood was diluted with 6 ml of PBS, phosphate buffer saline, of course sterile, 1x. And then this diluted blood was layered over in a separate tube. 3 ml of lymphocyte separation medium LSM from Imidacloprid was put.
Effect on a particular organ. So they are very wide in nature, highly distributed across their phenotype, and genotype to phenotype correlation can be sometimes very, very difficult to really understand what that particular disease is talking about. So if you look at the overall statistics of these rare diseases, then almost 80% of rare diseases have genetic origin, and 50% of those who suffer from rare diseases are actually children. And you will see that 30% of those children may not even see their 5th birthday, and 35% of the death in the first year could be due to the rare diseases. So it looks that these are orphan diseases. They are very, they do not affect too many people, but actually if you look at the statistics, this is quite devastating. And 2/3 of these are actually serious and disabled with improper or no treatment options available to this. And this actually has an enormous negative impact on the family well-being. The patients who are suffering, the whole family suffers, and they are, for a long period of time, they are just waiting for the diagnosis and the correct treatment options. And the whole diagnosis of the disease itself is actually very long and expensive. And often the frustrating part is that you never found an answer. So in part, because the genetic cause of at least is 1/3 of the rare diseases is not yet discovered, we do not know the genetic cause, that it is very difficult to hit upon the treatment options. So next slide, please. So when we are looking at the challenges, when we are trying to understand these research and the drug development, the problems is that we have very, very little information about a particular disease. So how do you know what the endpoint to examine? So most of the time, the doctors face this difficulty that this particular disease we do not understand, and we do not really correlate the phenotype of the patients because we do not have a clear understanding the pathomechanisms, the cellular models, the molecular biology. In fact, the natural history of the disease, we do not have a complete information about the whole of these diseases. So how to think about a clinical trial then? You know, it is such a big challenge that it is hard to ascertain how do we define a clinical trial. We really need a handsome number of patients to actually begin clinical trials. So it becomes a major limitations and a big challenge as to how do we identify a cohort of patients with which we can design a clinical trial and understand the disease. So we need to overcome this limitation in research and development that is caused due to scarcity of data, and we really need to come together and pull down all the data that is available with whatever different patients we have and make a consortium. So that is one, we need to promote an effective research and development to get more information, to provide more information, to provide more of the data analysis so that we can have some useful information that can help us go towards the understanding the treatment options. So earlier with most of these, as I said, is a genetic disorder, the diagnosis actually depends on the gene discovery. So with now current whole exome sequencing, most of the diseases are now identified. We know what all the genes are, where the mutations are. So you can try to overcome that problem either of diagnosis with the gene discovery, but still the major point is that you have to have a critical documentation of phenotypic variations in the same gene. What I mean by that you need to critically document the phenotypic variations that are happening due to the mutations in the same gene, and it is required that two unrelated individuals with the same rare disease can be correlated for their phenotypic variation. That is an essential requirement or an essential benchmark to categorize a particular rare disease. So phenotype of a particular rare disease. So you need to have that kind of a documentation to actually determine the genotype to phenotype correlation of unsolved patients with the rare diseases, and these databases need to be connected for matchmaker exchange. So now there is an MME program where you are saying matchmaker exchange program to really see a whole database that can be correlated with the genotype to phenotype correlation. So the development of the therapies will depend on the development pipeline of the drugs. So when you are saying that the therapy development, how many molecules you have. Now many companies and industries, they do not want to enter into the rare diseases because it is a highly cost effective area. You have very limited patients, and probably if they come up with a drug, there will be very few people who would be taking this drug and buying this. So eventually the cost of that drug becomes very high by the time it reaches to the patients. And you have, since very little number of patients, developing the clinical trial becomes very costly. And when you are bringing that product to the market, then there will be very limited number of patients who will be actually buying that drug. So, you know, you understand, so how you want to determine a research which is innovative and good enough to really see so that the cost effective is less, and you can provide the drugs at a lower cost to the population. So the problem that is the limited medical knowledge for these rare diseases and the greater patient cost associated with the drug development brings it a much bigger challenge to understand these diseases and come up with the treatment options. Next slide, please. So these difficulties, that the cost of developing a therapeutics that is compared to the size of the potential market, has been a challenge for many of the research scientists to enter into enter into study for the rare diseases, and you have to really convince them that there is a positive benefit risk ratio, given that so few individuals can participate in the clinical trial. So we really want to convince the people who are in the science that, see, there is a potential benefit. For cancer, I don't have to explain to anybody, because people say that there is a huge population which is getting affected by the cancer. So if one drug market, they will be able to make lots of profit. This is only for the patients who are really suffering, and there is a handful of patients who are suffering for these rare diseases. So what do we need to do? Next, please. So what we need to do is instead of doing different things, we need to do things differently. Basically, we need to be more innovative. We need to be more focused in the way we identify the target and study deeper into the research. Next slide, please. So we need to really understand what therapeutic options we have. Now we have, if you look at the different diseases, how do we approach? The small molecule drugs are often the most favorite of all the pharmaceuticals company, because they are easy to grow, and to bring it to the clinical trials level, these small molecular drugs are, I think, the most favorite among all the researchers, scientists, and even the industries. Then you have antibody therapies, where you develop antibodies and try to give them as therapies to the various treatments and disease treatments. Protein replacement therapies, these being the genetic disorders. Enzyme replacement therapy works that is caused due to mutation in a particular gene. Or you can have gene and cell-based therapies, where you use different cell-based models or pluripotent stem cells, which can help in the therapies towards certain genetic disorders which are affected by this. So next slide, please. So I'll just quickly brief what all successes we have. So the area is not that dull. I want you to give a message that a lot of work is being done towards the treatment of these rare genetic disorders. We have a lot of success in the area of many diseases, where you can look up to, get inspired, and try to address new of the rare diseases. So the small molecule drugs, why I said is a favorite one for most of the scientists, is because you can give them by different multiple routes of administration. You can give them orally, intravenous, intramuscular, whatever way you are interested in. Small molecular drugs are easy to handle. They can be easily administered to the patients, and you can very smartly control the dosing of those small drugs. So the dosage, you want that patients should not see the toxic effects of a high doses of a particular drug, and you have to limit the dosage. And with small molecule drugs, with the correct kind of formulations, you can actually control the dosing of these drugs, and most of the time they are stable. If they are chemical entities, you can work upon the stability of these molecules, and also you can scale up the synthesis of chemical compounds or the pharmacological compounds, and these most of these small molecule drugs. They are generally low cost because they are easy to manufacture, they are easy to manipulate. You can easily make even generics out of it, and you can target many tissues simultaneously. The tissue delivery is not a big problem with these small molecule drugs. So, so they are most of the time favorites. If for any disease you can identify a target for which you can identify small molecule drugs, then half of the battle is won. Then the challenges which are associated with the discovery of these small molecule drugs is you have to identify a right molecule that displays excellent pharmacological effect and pharmacokinetics, so that you have very few off-targets effect and you can avoid those kind of off-target effects, and be specific in terms of how this molecule is affecting your target directly. You can have an access to sufficient number of chemical entities, and sometimes across the chemical entities also you can find drugs. So for example, right now we are looking at some of the anti-diabetic molecules, chemical entities which could be of use in our neurological disorders. So that way it also becomes an advantage, but at the same time it can be a challenge where you have to how you have an access to a common pool of all the chemical entities, so that I can access maybe an anti-diabetic molecule for my own neurological disorder. So that becomes a limitations at time, that you want to have sufficient number of chemical entities so that you can explore more and more options. You need to have development of very stringent screening process to identify which of these molecules can be taken further for the drug development into phase 1, phase 2 clinical trials, so that you don't fall at that stage. So if your screening abilities are very good right at the beginning, there are chances that you will succeed. Otherwise, later 10 years down the line, you will probably get back to square one. So here, just I have listed 2 or 3 diseases like cystic fibrosis. We have ifacaftor and umacaftor, which promotes CFTR's trafficking and promote the folding. These are small molecules. They have cleared phase 3 clinical trials with FDA approval. And for lysosomal storage disorders, we have miglustat and aliclustat for Gaucher disease, which have approved clinical trials, and they are involved in inhibiting the biosynthesis of substrate, which for the enzymes which are lysosomal defective enzymes, and they are responsible for causing lysosomal storage diseases. Similarly, for Duchenne muscular dystrophy DMDs, we have ataluren, which promotes the stop codon reading through the dystrophin gene. So you have a dystrophin gene which is defective, and this particular compound helps promotes the read through the stop codon, so that you can still have the dystrophin protein formed, and that's how overcome the problem. Next slide, please. So when we look at the drug identification for these small molecule drugs, it will depend critically on the screening technology, development of chemical libraries. You have done a good ADME profile, and you have a whole high throughput systems. Next, please. So when we are looking at more of these specific changes, you have antibody therapies, which are well studied, long process, like 30-40 years. They have been there in system, phage display, transgenic animals. We use B cell infantilization and single B cell sorting to make the antibodies, which could be engineered. They could be full-length monoclonal antibodies, and also there are bispecific antibodies which can block 2 of the targets simultaneously. And at the same time, you can have fusion of the antibody constant region to the non-antibody related protein. So these are different strategies which are available when you are trying to design antibody therapies for a particular disease, and you can use any of these approaches to overcome a disease like Herceptin we have for breast cancers, and those are there. But here we have Actunizumab, which is targeting the complement protein C5 in the treatment of hemoglobinuria, and canakinumab, which targets cytokine beta, and that was originally used for rheumatoid arthritis. And similarly, we have now recent nanobody therapeutic, which is caplacizumab, which is for the thrombotic thrombocytopenia purpura. So all these examples indicate that you have antibody therapies available for certain rare disorders which can be addressed. Only problem here is the limitations are that you have the large size of antibodies can limit the tissue and the cell penetration, and that is why the recent work is on nanobodies, which can be considered as a good option. The second is because we are going to make these antibodies in the mammalian cells most of the time, you are going to produce, there is a high manufacturing cost. And like you would all have experience with the vaccines, Covaxin and these, you always have the injection site adverse effect, which could limit the patients to take up these antibody therapies. Next, please. So there are limitations with respect to the antibody therapies, but still we have a lot of success that can be considered for developing antibody therapies. Next, please. So the advantages, of course, lies with these is the specificity, because this is highly specific, and also the stability in vivo allows infrequent dosing regimens, because once you have an antibody treatment, once a month will be good enough to provide you protection for that disease or infection. Next, please. So the protein replacement therapies are also available for a lot of rare diseases where you are looking for some of the enzymes being replaced. Like if your disease is caused by a particular enzyme that is coded by the mutated gene, then you always go for enzyme replacement therapy. Only problem there is that you need to have a recombinant, good, robust recombinant protein production system, which could be bacteria, yeast, mammalian cells, or baculovirus. And our lab has developed dictyostelium also as a protein recombination system, where at a low cost, you can produce eukaryotic genes or therapeutic proteins, which can be used for drug development. So baculovirus is good because post-translational modifications can take place. And of course, mammalian cells are the most favorite ones, but they are quite costly if you are talking about the industry and the drug development. So all these expression systems needs to be worked out on the enzyme that you want to give to the patients for the gene recovery. And only thing you have to keep in mind is the delivery of these replacement enzyme, that too at the right dose and into the right tissues and the cells. And when we are talking about the neuropathies, these enzymes are too large to cross the blood-brain barrier, and that's why it becomes quite challenging to look for these kind of replacement therapies. But still, say because they are safe, they are preferred. If we ignore the high manufacturing cost of these enzyme replacement therapies. Next slide, please. So with current options, we have oligonucleotide therapies which have provided good success, and the antisense oligonucleotides, these involve a short strand of modified nucleotides that target RNA in sequence-specific manner. So when you are looking at inducing the target protein knockdown or the restoration, then they are the quite favorite antisense oligonucleotides or siRNA are the favorite therapy options. They have high specificity, reduced toxicity, with limited problem that is only associated with the delivery of these compounds. So the delivery vehicles and the conjugates, if you optimize and work it out, then you can lower the dosage and minimize the toxic effects that may be caused, any of the side effects or the toxic effects that could be caused due to the delivery of these molecules. So since these oligonucleotides are highly specific, they will be shutting down the particular target. It becomes very important that you identify the particular target. If you have identified good target for the disease, then half the battle is won. So it is important the right gene to be identified, and you knock down that gene expression using antisense oligonucleotides or siRNA and work out the proper formulations for the delivery of these molecules to the right site. So in 2018, the first siRNA approved therapy was observed for transcitrin-mediated amyloidosis, that was patisiran. That was approved in USA and got the approval from FDA. Similarly, you have nusinersen, which increases SMN mRNA transcript. It is used in the treatment of SMA. So these mRNA transcript are given that will increase the SMN mRNA transcript to overcome the defect. Similarly, if you have to correct the dystrophin, by exon skipping, if you see that there is a defect in the exon, dystrophin exon in the DMD patients, then you can give this eteplirsen, which is also a oligonucleotide therapy given to the patient. And recently for Huntington, this antisense oligonucleotide has been identified, which is RG6042, and that has got an FDA approval for treatment. Next slide, please. So when we are looking at these different therapies, I'm just coming to the last one before I really discussed all my research quickly. So the gene and the cell therapy based, we have to identify the expression of the gene that is has lost the function, then you can give that gene, particularly, for example, in SMA. In Huntington, we give RNAi to inhibit the pathogenic gene expression. And similarly, in ex vivo and other diseases, we can work out using the induced pluripotent stem cells. We can give to the patients to overcome the loss that is observed due to the cells. So I would scale that. I think I'm running short of time. And please, next slide. So our lab basically is focusing on some of these disorders. And here, when we see all these therapeutic options, then we have to look at the role of the patients. That is important here, because we do not have the right kind of drugs and treatments available. So the patients actually become your research partners, because they also help in the identification of the clinical endpoints, as well as you need to inform them with the consent documents and how the disease is going to progress. And you sometimes actually encourage all these patients to get recruited and participate in the development of the drug itself. And these patients can meet the regulatory agencies. These patient organizations are formed. They discuss and come up with the problems that they are associated, and clinicians as well as researchers can club and get the information about understanding of the disease and coming up with the whole drug development. Next slide, please. So my lab is working on a rare genetic neuromuscular disorder, which is actually characterized by proximal and distal muscle weakness. And it is basically affecting anterior tibialis muscles, but spares the quadricep. So it is rare because, click please, because if you see the different countries, you will see that in India, we have more than 50 patients, and now I would say more than current data is more than around 200 patients have been identified, and that's why it has been categorized into a rare genetic disorders. And this disease is often seen at the onset of adulthood, early adulthood. Until that age, these patients are all normal, but eventually become wheelchaired in an average span of 12 to 15 years. So pathologically, these patients are characterized by muscle atrophy, which is the presence of, pathologically and clinically, they are also seen with the presence of lymph vacuoles and protein aggregation. The inheritance is autosomal recessive, compound heterozygous, as well as homozygous mutations have been seen, but no null mutations have been observed. So how do we diagnose these patients? These patients are diagnosed with the biallelic mutations in the gene GNE, which is the key sialic acid biosynthetic enzyme that, if defective, cause hypercellulation in the cell, and patients suffer from various phenotypic characterization, which our lab is trying to understand what all is happening in this disease. So far, there is no treatment, no cure. Only thing is we give sialic acid supplementation to these patients, and there are some molecules where we have looked at sialic acid modulations for effective gene delivery. And the phase 3 clinical trial of a modified version of sialic acid has also failed last year. So there is a click, please. So when we see these patients, they have a different this is how their legs and the hands they look like, and eventually they become the wheelchair. So these really need a good treatment because we do not understand much about the disease. Next slide, please. So these, as I said, these are caused due to the deficiency of sialic acid, because the gene that is causing the disease is mutated, which contribute to the key sialic acid synthesis. And these are 9 carbon backbone negatively charged hydrophilic molecules that actually occupy the terminal position on the cell surface glycoproteins and glycolipids, and they are involved in various cellular processes including cell addition, motility, macromolecule recognition, determination, and so on. But they are widely distributed in all the tissues of animal cells. But in our disease, we are seeing only the skeletal muscles being affected, and why it is only the skeletal muscle, we do not understand. So sialome is one of the way by which the total array of cyclic acid types and lymphocytes can be determined, and we are trying to see how these the variation is observed with respect to the sialic acid. Next, please click. So when we are seeing the biological roles of glycans, you have lots into virus, host, pathogen, intrinsic recognition, extrinsic. So all the immunological role, these sialic acids are involved, and they are known to perform key roles in the cell adhesion migration. Next slide, please. So how, I'm sure being graduate students, I just thought I'll quickly go through what is the importance of sialic acid and how it is added there. So when the protein is being synthesized in the ER, so you have where ribosomes, the protein synthesizing glycosylation process, which is going to begin in the ER. And as the protein moves into the Golgi, you see various kind of glycosylations that are taking place, O and N-linked glycosylation. And particularly in N-linked glycosylation on the molecular backbone, you see in the ER, there are additions of glucose residues, as well as it migrates to the cis medial trans Golgi, you will see manosidases which will be working, there will be glycosidases, and the whole, I'm sure you know the pathway of this. And in the trans Golgi, in the end, you will see that sialic acid is added as a terminal residue. And once the sialylation takes place, the protein is expressed on the cell surface, where it is now going to perform all the cellular function. Next slide, please. So when you see the sialylated protein, then you see the various functions which are associated with the sialylation are being performed, and the sialic acid biosynthesis that begins in the cytoplasm involves the conversion from glucanac to mannanac and then phosphorylation, which are the 2 key steps that are regulated by GNE, and it has an epimerase and the kinase domain that will regulate or cause, if there are mutations, lead to GNE myopathy. And the whole pathway will guide the addition of sialic acid to the protein and then expression on the cell membrane. Next slide, please. So there are various disorders which are associated with the defect in GNE and the causes. Next, please, I can skip this. So and the role. So the gene that is there, click please. So GNE is, UDP N-acetyl glucosamine 2 epimerase manosamine kinase, and it has an N-terminal epimerase and a C-terminal kinase. Next, please. So next. So we are looking at how these genes are regulated. They are caused by oligomerization and phosphorylation and feedback inhibition by CMP-NANA. And also you have epigenetically, they are regulated by methylation of the promoter region and the phosphorylation by PKC. This is just the known mechanism that is there. Next, please. So these different muscular dystrophies, we know the cause, we know the problems which are associated, we know what genes are there. But click please. But for this, we do not know what are the molecular mechanisms that are associated with GNE myopathy. Next, please. So the mechanisms that our lab is trying to understand why this GNE myopathy is caused. Click. So there is a possibility of mitochondrial dependent apoptosis because of because of the reasons, the science that has shown that there are impaired apoptotic signaling associated in these cells, and microarray studies have shown changes in the mitochondrial proteins, and aspase activation has been observed. Next, please. So there is a problem which is associated with the hypocellulation. We know that. And click, please. Keep clicking, I will go through quickly. So hypocellulation is there, and the ER stress and the UPR pathway that can be a major cause of this disease. These are the cellular mechanisms that we are looking at because of some of the earlier studies that have been performed in the area that shows that there could be changes in the upregulation of chaperones. Next, please. And accumulation of amyloid beta, and we see a lot of autophagic vacuoles in this particular disorder. Next, please. So we are working on trying to identify the alternate roles of GNE myopathy besides sialic acid biosynthesis, as we are seeing that there are changes in the organization of cytoskeletal network, there could be cell apoptosis, there could be an effect on ER stress and UPR pathway, and other cellular processes. If there are any, then we need to identify the novel proteins. Next, please. So all these have been worked. The first challenge, as I was discussing earlier, is that you need to identify the cell-based model to initially begin and trying to address towards the therapeutics. And certain leads from our lab has identified that there is hypocellulation of beta-1 integrin and the IGF-1R, as well as altered cytoskeletal organization with the changes in the F-actin polymerization, as well as changes in the ER stress levels due to the PERK pathway and IRE1 pathway and mitochondrial dependent apoptosis. So we have identified few of these cellular features and trying to develop the therapeutics with respect to the key molecules that are identified as an effective molecule and trying to address the drug molecule. So we have cytoskeleton apoptosis, ER stress, and protein aggregation to affect the pathomechanism of this disease, which might contribute to the rimmed vacuole formation. Next, please. So these were some of the success stories that we have, because when we started the lab, we made this HEK cell-based model with the pathological relevant mutations of GNE being expressed. Earlier at that time, we knew only Japanese and Israel. Now we have Indian mutations known, and we have a system where we have the cells which are expressing these recombinant mutant proteins. We can try to address various cellular phenomena. And then we have now successfully developed homologous recombinant cell line with the deletion in this gene in the exon, which is coding for epimerase, so that now we can address and see what is happening in the muscle phenotype. Next, please. So we have filed now this patent, and if you can see here, you can see that with actin, you can see striations in the muscle cells. But when we are looking at the mutation or the cell line which is knocking out GNA, you lose these striated patterns and actin is totally disorganized. So we have a good success over here, and this seems to be a good model to now test all our drugs and the molecules that we can try to see how they can be beneficial for the cells where you have a defective skeletal muscles. Next, please. This is, we have looked into the cytoskeleton pattern. We are trying to address the cytoskeletal organization, and we have looked into beta-1 integrin pathway where the focal adhesions, the Rho A, and the signaling cascade is affected. And Rho A we found to be upregulated and cofulin downregulated. And we have identified the key molecules which can alter the activity of these. So you can refer to these papers if anybody is interested to go how we have studied. Next, please. Similarly, the apoptosis, because we were looking at the loss of muscle mass, so there is a loss of muscle cell death. So how you can overcome that muscle mass loss. We were trying to identify if we can rescue the cell death which is observed in these muscle cells, and we found that IGF-1R signaling pathway was good enough, and treatment with IGF-1 balanced out the cell death that was observed, and we could see that lot of cells were moving towards the survival, and you can rescue the cell death which is there. So it looks like a promising treatment, right? If we treat with IGF-1, and we observed that in NIH, a lot of patients were given IGF-1 growth factor treatment, and it was helping these patients. Next, please. Similarly, we have identified with the ER stress some of these molecules where we can determine that how the ER stress redox potential is altered in the various mutant cell lines, and you can see the ratio between these blue and the green ratios that were altered in the mutated cell line, and treatment with IGF-1 again lowered the ER stress that was observed in this cell line. Next, please. And protein aggregation, as I said, beta amyloid and these things, they were there, and we found that aggregation to be there. And the chaperone HSP70 activity was found to be associated, and the modulators and effectors of HSP70 again can give us a good target leads, drug target leads to identify HSP70 modulators, and that can be developed into a drug candidate. Next, please. So this was the actin, apoptosis, ER stress, and all HSP70. These are all the cellular molecules, cellular pathways that have been identified, and looking at how these molecules are going to alter these pathways, we can come up with the drug molecules. Next, please. And the unanswered question for this development is why mutation in DNA affects only the muscle cells, though it is there in other cells and tissues, and how this complete process of protein aggregation is initiated in DNA mutant cells so that we can overcome and inhibit this protein aggregation process itself, and why is it on the adult onset as it is the hormone control. So these are very intriguing questions to understand the pathomechanism of the disease. Next, please. So from the whole this call, we are saying that how you are going to differentiate the stem cells to muscle cells. Can you do the gene editing? Can we identify some of the anti-diabetic molecules? Basically, how we can approach and improvise upon the drug development. And of course, all this work could not have been completed without the proper support of my lab and the hard work of all the research students who have contributed tirelessly to the understanding the whole very difficult problems in this particular rare disease. So I thank all the JNU faculties as well as my lab members who have tremendously contributed to the projects that we are running. I'm sorry for running through too many things. I hope I have not taken much time, and I welcome all the questions. Thank you.
Any questions? So, thank you, madam. What are rare diseases, their challenges, difficulties, therapeutic options, limitations? All the topics are very well explained by Dr. Arya. Thank you, madam, for such an informative lecture. It definitely would be very helpful to for everyone.